I'm going to turn it over to Jill to introduce our speaker today.
Thanks, Craig. Well, it is my very great pleasure to introduce Dr. Kayri Havens, who is visiting us today from the Chicago Botanic Garden. She is the Medard and Elizabeth Welch Director for Plant Science and Conservation at the Chicago Botanic Garden. She completed her bachelor's and master's degree at Southern Illinois University. Then she went on to do a PhD in biology at Indiana University, and she's associated with many international professional organizations, including IUCN, the American Public Garden Association, Botanic Garden Conservation International, and many others. And her research is broad. It encompasses reproductive ecology, conservation of plants, ex-situ collections, methods development for conservation, and she works a lot as well with citizen science. And when we met way back in the day, pre what we're living in now, in a beautiful day in South Africa, in Cape Town, we enjoyed some very nice wines and similar love for Welwitschia, which I was very jealous because Kayri was going to see Welwitschia in its native landscape in the Namibian desert after the meeting we were at. But we enjoyed very common interests in seeds and collecting seeds and seeds for restoration. So I'm really excited to hear the talk today and to hear what she's been working on recently. So thank you so much for joining us today, Kayri.
Thank you, Jill. Wonderful to see you again. I wish we were in South Africa enjoying a nice Sauvignon Blanc, but alas, here we are. I did make it to see Welwitschia after our meeting. It was something on my bucket list, so I was very happy to see that wonderful plant, wonderful and weird plant. So I'm going to talk today a lot about different things that botanic gardens do to conserve biodiversity. And the talk will really be in 4 parts. The first focusing on ex-situ conservation, then on research on rare plants, then on restoration and seed sourcing, and finally on public engagement. And these are activities that all botanic gardens do to some extent. And I'll give examples from our own work, but please know that this is common in the botanic garden world. And if this is the kind of thing you like to do, I would encourage you to think about a career at botanic gardens. So I'm going to start talking about how we keep species viable through ex-situ or off-site conservation. This is the kind of conservation that happens at zoos and botanic gardens. And the history of ex-situ conservation is fairly long. Botanic gardens have been repositories of plants for academic study throughout their entire history, dating back to the 1400s. But the first time we thought about gardens serving as conservation organizations was back in the 50s. Botanist named Cugnac in France talked about the conservation garden and how we could support wild plant diversity. And in the 70s, we thought more about botanic gardens as arks, where we would temporarily hold plants and then get them back out into the wild. And finally, more recently, we really think about ourselves as partners in ex-situ conservation, that we work hand in hand with people who are doing land management and support species recovery in a variety of ways. So there are about 3,300 botanic gardens in the world, holding over 2.5 million accessions or collections of wild plants, representing about 1/4 of all plant diversity. Roughly 120 species are extinct in the wild and exist only in botanic gardens. And for many plants, what's left in gardens is much greater than is in the wild. Many of those plants are being reintroduced. And we work to support the global strategy for plant conservation, and in particular, Target 8 of the strategy aims to have 75% of threatened species in ex-situ collections and 20% in reintroduction programs. So the way we do this for most plants is seed banking. In this picture of our seed bank at Chicago, most plants have seeds that can withstand drying and freezing. And if that's done well and the seed is harvested at a time when it's mature and viable, we can keep these seeds frozen for, anywhere from 20, 30 years to 200, 300 years. Based on artificial aging studies, we think 200 years is the average that plants or that seeds remain viable when properly stored. But there are about 30% of seed plants that have seeds that can't be stored frozen. They're called recalcitrant seeded species, or sometimes they're called exceptional species. And they need some sort of other conservation method. And that could be a living collection, that could be maintaining little explants in tissue culture. It could be taking a part of a plant, freezing it in liquid nitrogen, and then bringing it back to life later. But mostly it's these living collections, botanic gardens and arboreta, where these plants are maintained. And many of the plants we care about deeply, like chocolate, like avocado, are exceptional species and need to be maintained in living collections. We've done some work looking at how many plants we need in these collections to capture genetic diversity. And usually our goal is 95, which is this kind of faint dotted line here. And this is looking at a number of different tree species, and the lines of similar color here, like all the blue lines are oaks, and the pink lines are hibiscus. So same genus if they have the same color. And you can see there is variation in how many plants we need to collect in order to capture genetic diversity. But if we look at this 95% line, it's anywhere between about 25 plants to around 75 plants per population in order to capture adequate amount of genetic diversity. And for big things like oaks, that's a lot of trees to try to maintain in a living collection. And we also know that there are many more challenges with living collections, and this applies equally to both plants and animals. It's riskier because if you have those plants and animals together in a collection, they can hybridize. They lose genetic diversity either by drift or selection to their captive or cultivated environment, or inbreeding. They can adapt to cultivation, they can get sick, they can fail to reproduce, they can die. So we don't like to use living collections if we can avoid it. The zoos have faced this for decades, and they have developed a lot of tools that track the pedigrees of the animals they have in their collection, and they manage those animals as a single metapopulation, and they use software to decide who to cross with who in order to retain as much genetic diversity as possible. We're testing this approach now in plants. We have a grant from Institute Museum and Library Services to do this. And, plants face many of the same challenges as animals, and the tools that the zoos are using may be quite successful. So the first plant that we're testing this on is called Brighamia insignis, or cabbage on a stick. I don't know, you can't really see it here, but it's got this kind of succulent trunk and then a big group of leaves on the top. Much like many zoo populations, the collections we have in botanic gardens have relatively few founders. They have been held in gardens for multiple generations. Their pollinator is extinct, and so they require hand pollination. And we've seen decreased pollen production in Hawaii, which is where the core collection of this species is housed, suggesting that inbreeding depression is starting to take a toll. So this is the pedigree we produced for this. And all you really need to know is here are the founders, the original plants collected from the wild. Most of them were from a single site here, the Nepali coast of Kauai. And one individual was collected from a different site. And if the box is yellow, those lineages have been lost. So those are plants that have died. And let's see here. This is microsatellite study looking at this same species. And at National Tropical Botanic Garden, you can see this is a structure diagram. You can see that it's overrepresented in this yellow lineage and underrepresented in the other two. Fortunately, at botanic gardens in other parts of the world, we have just the opposite pattern. So we have a lot of diversity that's been lost from Hawaii, represented in collections in other places in the world. And the software that zoos use is called PMX, which stands for Population Management Software. And it can determine which individuals should be crossed, as I mentioned, to maintain diversity. And so we've looked at this in Brighamia, and it's suggested crossing a plant from Waimea Arboretum with a plant from the Botanic Garden in Switzerland. And so we have made the pollen transfer and are implementing many of the crosses now recommended by PMX. The software also tells us which plants are redundant in a genetic sense, so overrepresented in collections, and those might be ones that we use for education and outreach, or we try some reintroduction methods because we don't need to worry so much about losing those individuals. So I'm going to move on to talking about research on rare plants. This is one of my favorite field sites. This is in Door County, Wisconsin, and this is where I work on pitcher's thistle, or Cirsium pitcheri. And Cirsium pitcheri is a dune species. It has to live on sand dunes, and it lives mostly around Lake Michigan, a little bit over on Huron as well. Federally listed as threatened and found only in the Great Lakes, Western Great Lakes region. We've been working with this plant, we call it SIPI for Cirsium pitcheri, for more than 20 years. We've been banking seed, we've reintroduced it at a state park called Illinois Beach. We've been doing long-term demography work, and more recently understanding its role in the pollination network. So I'll focus on that last question. Do rare plants provide significant resources for pollinators? We're asking this in this species because it is fairly rapidly headed towards extinction. It is being impacted by weevils that were introduced to control weedy thistles, and they're having a really bad impact on this species. And so we're wondering what's going to happen as populations are extirpated to the other species that depend on it. And this shows plants blooming on the sand dune at the Ship Canal site, the photo you saw earlier, in 2016, and how many insect taxa we collected from those plants. And so early in the year, we have a little Arabis bloom. We didn't find any insects visiting it. Then Cirsium, 35 different species collected at a single site in a single year. And then a little later in the year, some of these other species, including common milkweed, which we think of as a pollinator magnet. But we only found 10 species of insects visiting milkweed. The second most visited plant on the dunes was a sedum, which is invasive. This is a non-native sedum. We think part of the reason this plant gets so much attention from the pollinators is that it blooms at a time when not much else is blooming on the dunes. So this is a pollination network diagram, which shows interactions between plants here on the bottom. And I know it's too tiny to read, but I'll point out the important bits. And then insects up here on the top. And the wider the bar, the more interactions that plant has with different insects. And so this is Cirsium pitcheri here, the widest bar, interacting with the most insects on the dune. And the most common species of insect that we find on the dune is a bumblebee, Bombus impatiens. So we modeled what it would look like if we lose Cirsium pitcheri at that site. And unfortunately, I don't know if this is blocking the top of the slide for you, but I have a Zoom panel that blocks the top of the slide for me. So what it says behind that panel is, I think 9 of 27 pollinator species on the dunes would be lost. So without Cirsium pitcheri, we may see the local extirpation of many insects as well. So now I'm going to move on to some of the restoration work we do, and including our seed sourcing work. So we have a large restoration debt in the US, particularly in the Western US. We have millions of acres that need restoration. Over 10 million just on BLM lands alone have either recently burned or are infested with weeds, primarily the cheatgrass shown here. But restoration is seed limited. Bureau of Land Management is the largest seed purchaser, probably in the world. In a bad fire year, they need to purchase up to 7 million pounds of seed a year. And because they have such high volume needs, they can't always get what they want. They can't always get what they need either. There are a lot of ecotypes they can't get, and about half the species they purchase are non-native because they can't even find native species. So to deal with this, several federal agencies in the US developed a national seed strategy in 2015, and we are working on part 2 of that right now. And it outlined a series of tasks that needed to be completed in order to have the right seed in the right place at the right time. And the cost of implementing the strategy, they did a business plan that looked at that. It's about $360 million. And one of the really important tasks, which is underway right now, is conducting a national assessment of seed need and availability. And that work is being done by the National Academies of Science with the final report due out this fall in September. So this is just kind of an example of the problems BLM in particular are facing, that when they want to restore areas that have burned or are infested with cheatgrass in the Great Basin, shown here in green, what they can buy on the commercial market are mostly grasses, mostly cultivars of grasses selected for usually forage quality. Most of the provenances of those cultivars are where these stars are. So mostly from the north where it's cooler and wetter, which is not what you want to do under climate change in the Great Basin, where it's predicted to get even hotter and drier. And there are very few wildland collections of woody plants and forbs for that area. And we know it's really important to have the right seed because of many provenance studies. And this is just one that I'm showing you here, excuse me, done by Alan Sands over a very long period of time. He collected Wyoming big sagebrush back in 1987 from all the locations shown here with dots. And he grew them in a common garden in Glens Ferry, which is this red dot right here. And then he went back 23 years later and looked at what was still surviving. And virtually all the accessions had lost 50% or more of the plants. But the Glens Ferry, the local accession, was still completely alive at that time. One of the things that we need to think about when we're looking at restoration and seed needs nationally is that there are regional differences in both seed need and kind of the process of restoration. So out here in the West, we have very large scale projects, often done sowing the seed by helicopter because they're doing such huge areas of restoration. And that need is driven by unpredictable disturbances like wildfire. And the seed transfer zones shown in this image, different colors are different seed zones. They're much smaller in the West, driven by all the elevational gradients out there. And how plants move in response to climate change is much less predictable, also because of these elevational gradients. Whereas we here in the Midwest have smaller scale projects, often predictable, often agriculture land coming out of use and being restored, so that we can proactively develop the seed that we need for that particular restoration. We have larger seed transfer zones, and we know from species distribution modeling that plants are largely moving north and east in our part of the world. So I'm going to talk about some of the restoration research that the garden's been doing in the Tallgrass Prairie. This is a highly endangered ecosystem, a highly diverse ecosystem, and it is where restoration ecology was born, with Aldo Leopold doing the earliest prairie restorations about 80 years ago now. And when we're doing restorations, we know that we want diversity, but that can be measured a lot of different ways. Species diversity, genetic diversity, phylogenetic diversity, which is how many branches of the tree of life are represented. Phenological diversity, do we have species that bloom across the entire season? And functional trait diversity, do we have plants that do different things? So woody plants, grasses, forbs, big leaves, deep roots, etc. So this is work by Becky Barak, a former student and now colleague at the garden. And she asked, how do restored prairies differ from remnants? And in particular looked at species richness and phylogenetic diversity. And here's the tree, the phylogenetic tree that she used. And the most common species in the prairie are grass, sedges, mints, roses, legumes, and asters or sunflowers. And I want to have you look at this panel first, this upper right panel, which is species richness native plants. And for each of these panels, the green box is the diversity of the seed mix that went into the restoration. The purple box is the diversity that's standing in the restoration. And the comparison, the blue box, is remnant prairies. And so you can see for native species richness, kind of what went in is what came out, and both are considerably less than what you see in a remnant. The same is true for phylodiversity. So how many branches of the tree of life again? What went in is very similar to what came out and much less than remnants. The one measure that differed a little bit in its pattern was this mean C, or coefficient of conservatism, which is a measure of rarity or conservativeness of a plant species. And you can see we're sowing in a lot of conservative species, but we're not seeing those come out in restorations. And so there may be a lot of species we're wasting money on that we're trying to restore that just aren't working for one reason or another. When Becky looked at what was missing from restorations compared to remnants, it's probably families that won't surprise you. Lilies, orchids, and a lot of parasites and hemiparasites. These lilies and orchids in particular are very hard to grow, very hard to establish in restorations. And the hemiparasites also have been fairly difficult to propagate and establish. And then some, like dodder, a parasite, nobody is even trying to grow it because it's not something that the restoration community perceives as valuable to be restoring. So next, we talk a little bit about phenological diversity. And remember I talked about Cirsium pitcheri and why it was so important, because it bloomed in the middle of the season when not much else was blooming. We see the same pattern in prairies. This is work that dates back to Edgar Anderson. He actually did this work in the mid 1900s, but didn't publish it till the late 1900s. And he looked at when plants bloomed, and you can see two peaks, a spring peak and a late summer peak. And then this period in kind of June, July, and this work was done in Missouri. And he called that the green lull, the time when nothing's in bloom in the prairie. And so we looked at seed mixes that are recommended for pollinators in Illinois under the CRP program. And we looked at dozens of mixes. Many of them had many more species than shown in this particular mix. But what we found consistently across all of them is they were dominated by these late summer and fall blooming species and missing the spring peak altogether. And when you think about pollinators, that you need to support them throughout the growing season. And so you need those spring blooming plants. And they're just not available in seed mixes. And here are some of those spring bloomers that we'd love to see incorporated into seed mixes. Again, we have some hemiparasites, Lithospermum also. We have some things in the lily family. And then the violets are also very hard to propagate. So missing some important species in our restorations. And so kind of, if you look at this overall, are we meeting our diversity targets in our prairie restorations? And to a large extent, we're not. So if you look at a remnant, and then what's available commercially to sow into that remnant, the restoration species pool is considerably less diverse. So only 54% of prairie taxa are in the trade. And once you sow those in, there are a number of factors, genetics, biotic factors, abiotic factors, land management factors that mean the restored community is even smaller than the reference, and many species then are kind of relegated to the dark diversity. What this means, I think, is our restoration practices are really shifting what prairies look like and how they function. And this is something we need to do better on. So when we think about seed sourcing, so I've talked a lot about, what species and how many species make it into restoration. Well, once we've picked our species, we also want to think about what provenance of the species we use. And there are a lot of questions about seed sourcing and provenance. And they range across this entire spectrum from the kind of traditional restoration I was just talking about to roadside use, prairie strips, green roofs, pollinator gardens. Everyone's asking, how far away can I go and collect seed and use it in one of these applications? And there are a lot of answers to that. Some people look at the USDA plant hardiness zones or elevation shown here, or a combination of that shown in these provisional seed zones developed by the Forest Service, or something that's even more conservative. And this is for the forest preserves of Cook County, where I live. They'd really like to just use seed from Cook County. They don't want to go outside of the region at all, and their preferred seed zone is the county itself. And this leads a lot of people to ask the question, well, what's happening under climate change? We've always thought local is best, and there was a lot of reason to think that. And, I showed you with that Wyoming big sage example that many, many common garden studies have led us to think local is best. But that kind of assumed that the climate was stable and the environment was stable. And we know that's not true. So should we be taking climate into account as we look at provenancing strategies? And so here's, the local is best idea, and a lot of people still use that. Some are focusing on most of their seed locally, but getting a few from outside the region, or a very kind of equally distributed across the region provenancing strategy, or two predictive strategies. One where you collect the seed from the environment that you're going to experience 10, 20, 30, 50 years down the road, or this climate adjusted provenancing, which collects from a variety of populations along a gradient that is headed towards that climate you're going to experience in 10, 20, or 50 years. And so a lot of these different strategies are being tested, and we're testing some of them too. But there are reasons why you wouldn't want to use seed from a variety of populations. And one of them is different populations can have different ploidies. And this is quite common in the aster family and the grass family, which are two of the big families that, particularly in tallgrass prairie restoration, that we pull from. And so when we looked at restoration species in the US, 39 of 113 of them had interspecific ploidy variation. And we really need to be careful with those that we aren't mixing populations of different ploidy levels, either in production or in outplantings. When we're thinking about predictive provenancing, this is some work done mostly by a former postdoc named Shannon Still. And we looked at how climate change was going to affect species ranges. And these are rare species that we did this work on. But the vector shown here goes from the midpoint of the current range to the midpoint of the future range. And we did this comparing 2020 and 2050 climate projections. So you can see, I mentioned earlier that how plants in the West respond to climate change is a lot less clear than how plants in the East are going to respond to it. And plants in the East are going to be moving, or going to need to move if they're going to stay within their preferred climate envelope, pretty long distances, mostly north and east. So what does all this mean for provenancing? And so we kind of divided this into species characteristics that affect provenancing and site characteristics. And we think we need to be more conservative, so have pretty small seed transfer zones for plants that are narrowly endemic, that have ploidy variation, that we're uncertain about their taxonomy. There might be some cryptic species there, and that have little long distance gene flow. And the sites that we need to be concerned about are ones that have, always been isolated, high quality habitat, very stable landscapes that haven't changed over time, near high quality sites and larger sites. And we can go bigger, we think, on plants and sites that have the opposite characteristics. So this slide, I've been working a lot thinking about what would a native seed system look like in the US, a robust native seed system look like. And I think there's kind of 4 tiers that we need to think about. First, in terms of public land policy, we need to protect intact natural areas. Many areas are being over-collected, so we need to protect seed reservoirs. We need to mandate some areas that are done, that the restoration is done really carefully. So some of those sites that I talked about on the last slide that are high quality, we need to really focus on restoring those with regionally sourced native plant only material, unlike a lot of the restoration that's done, particularly out west. We need to have botanists determining seed needs, and we need to communicate those needs to the production sector. And I think native seed is inherently a local product. And so we need some regional seed hubs that are collecting, cleaning, and storing seed. Native plant-focused plant material centers that grow, learn how to grow that seed and liaise with the private sector growers. There may be some species that are never profitable for the private sector to produce, and so we may need the public sector to work on those. I think we need to focus on more proactive restoration to even out supply and demand by basically increasing demand. We need more cold storage for big lots of important workhorse species. Restorations can be implemented more iteratively, adding species over time, which allows us to really get the source that we want for many species. And we need to do these as experiments so we can learn from them. And lastly, we need partnerships with scientists who will help the restoration sector delineate seed zones, look how climate change is factored into sourcing, look at how best to sow those seeds, and synthesize the data from the successes and failures. So that's my vision, hopefully for a robust native seed system, and working on policy angles to get some of this implemented. And that brings me to this idea that engagement and advocacy are really needed in restoration and plant science. So in order to build the political will for some of these changes to happen, we need people to understand that plants aren't optional. And we need work like this to be better funded. And one of the ways we're hoping to convince people of the necessity of work like this is by engaging them in science. So this is one of our community science projects called Budburst, which brings together scientists and citizen or community scientists to uncover the stories told by plants and how they're affected by climate change. Oops. Oh, had that backwards, sorry. So Budburst focuses largely on phenology, and that's the timing of plant life cycle events, like when do they break bud, when do they flower, when do they leaf out, when do they fruit. This example from maple, and I can tell you because my nose is running like crazy today that the red maples are blooming here in Chicago. So Budburst has been going on for now 13 years and has well over 100,000 observations, and we have more like 16,000 participants at this time. And not only do we track phenology, but we have specific research projects that we do using the Budburst platform. And one of these that's running right now is the question of nativars and what resources they provide for pollinators versus the wild type or true native species. And so we're doing this with a number of taxonomic groups, including Black-eyed Susan, where we have gardens planted with the native plant and a number of cultivars of that native plant. And we ask people to observe phenology and pollinator visitation of those. And so this is important because cultivars can vary, and a lot of things that are really important to pollinators, like color and scent and phenology. And these different types of columbine shown here. Here's a double columbine. And when we double petals in a cultivar, we're usually doing that at the expense of stamens, and so this flower probably lacks pollen. So here's some preliminary results from Penstemon digitalis, or foxglove beardtongue, with the wild type and 3 cultivars. And this is work that my graduate student Emilia has been working on. And this is number of pollinator visits on the y-axis, and the wild type you can see is the highest. There's one cultivar called Pocahontas that is similar, but the other 2 cultivars have far fewer pollinator visits. Budburst, if you've used it in the past, we've just undergone a number of tech updates. So we have a brand new website, and just this week, our mobile apps came out, so you can now have this on your phone and super easily track phenology and pollinator visitation on any plant of your choice. There's also species recognition software in here, powered by iNaturalist. So you can take a photo of a plant, iNaturalist will tell you what species you're looking at, and then you can record information on phenology or pollinator visitation for budburst. So I want to leave you with the idea that science alone won't save a species, that we need to be really active in policy and legislation as well. And some of the policy and laws I've been working on include the Global Strategy for Plant Conservation, the National Seed Strategy, the National Academy of Science Assessment of Seed Needs and Availability that I mentioned, and lastly, the Botany Bill, which I haven't talked about, but this is a bill that we created, we wrote with a number of other botanical groups to do 3 main things: fund needed research in plant science, encourage students to go into botany and land management as a career, and we do that with a student loan forgiveness program, and to build a market for native plant materials by strengthening the preference policy for the use of native plants on public lands. The status of this bill, it was introduced in the last 2 Congresses. That administration was not particularly favorable for moving a bill through that focuses on conservation. We're about to reintroduce into this Congress, and we hope maybe we'll have better luck. So I can certainly keep Jill posted as soon as it's reintroduced and has a number. And you can, if you support this, contact your senators and representatives and tell them why you think it's important. So I'll leave you with the idea that plants need us, and we need them too. We can't live without them, so we need to do our best to keep them viable and on this planet. And I have to acknowledge a whole bunch of colleagues and students that helped with many of the studies that I mentioned today. And thank you for listening, and as well as thank our funders. So with that, I'll stop, and I'm happy to answer any questions.
Thank you so much, Kay. That was fantastic.
It's a whirlwind tour.
Oh my goodness. I'm so excited that the botany bill is gonna go forward again.
Yeah, fingers crossed. We'll see it soon. So I see Lionel has a hand raised.
Yep. So yeah, first, the great talk. I really loved it. I actually had a questions. The scientific name just slipped my mind right now, but for the cabbage on the stick.
Mm-hmm.
You showed that map with like the different lineages, and I was just wondering whether, I mean, it's a clear distinction between lineages from like Western US and Europe and the most Western part that I don't remember the name right now.
Mm-hmm.
I was mostly wondering whether you knew whether those different lineages like represent really like geographical lineages, mostly like are those really like representative of the distribution of variation within these areas, or is it mostly the consequence of some genetic stochasticity, or maybe are those different populations like potentially adapted to those areas? Mostly, I guess in the end, my question is, would it be wise to consider crossing those different individuals to get like a big resource of genetically diverse individuals?
Yeah, so I may not have been completely clear when I described that. So cabbage on a stick is a narrow endemic. It's only found on Kauai, the Kauai island of Hawaii, and it's now extinct in the wild. And so the differences you saw between plants that are held in Hawaii, US, and Europe are changes that have happened during cultivation, or are the fact that we sampled some diversity from the wild that was different, and we have that in our collections, and it's underrepresented in Hawaii collections. So yeah, those aren't different kind of natural lineages, those are cultivated differences. Does that make sense?
Yeah, that makes sense. Yeah. Thank you.
Sure. Jacob?
Yes, thank you so much for your presentation. I must apologize in advance of asking this question, as I wasn't able to make the full meeting and full presentation as I had prior work obligations. However, it sounds like you're doing wonderful things and pushing forward in the political arena with this work, which is great. And that makes me wonder to what degree are tribal nations being consulted, and indigenous folks as well, in all your work in arenas, and like what that looks like, and if it has been a thought. And I'm sure it probably has, but I was just wondering if you could speak to that a little bit too.
Yeah, so particularly for the national seed assessment that NAS is doing right now, there's good tribal representation. So we have a member who works with tribes throughout the West on their seed needs. And we're also surveying as many tribes as we can as part of that. So we are taking into account their seed needs and what they would like to have available for restoration of their lands. And the same thing I think can be said for the national seed strategy. There was a lot of tribal consultation there as well. So
awesome. I'm really glad to hear that, because, you know, as you well know, that just has not been the case in the past. So that's awesome to hear. No, so that also brings me to my next question too. Are you working with the plant material centers with the NRCS for some of this work, just out of blatant curiosity? Or
yeah, it's funny, we just had a meeting with them yesterday. So right now, most of what they focus on is cultivar, and also a lot of non-native material. There's a desire to get them much more engaged in producing native plant material. They're under congressionally mandated staffing caps right now. And so we're working on changing that so they can hire additional staff that would focus on native plants.
cool. Thank you.
Looks like Marinas is next, and then Annika, and then Jess.
Yeah, hi. Thanks. I thought it was an excellent talk. I have in the past worked with plants my field is wetlands, but I've worked with plants that people don't like. And like your CP, do you have the problem that you put them out in places and people destroy them because it's a thistle?
I've had a number of people ask, because I do my work in very public parks. They ask what I'm doing and why do I care about a thistle? And I take the opportunity to tell them what a wonderful thistle it is and how important it is to pollinators, and it's native and it's not invasive, and it's actually federally listed as threatened. And so I try to engage them in what's interesting about it. But yeah, of course, people ask, why would you save a thistle?
Yeah. Yeah, I've had it where I used stinging nettles in the Netherlands for monitoring purposes. And we would actually plant them and then retrieve them. And I had to go through hoops to explain that, you know, we would remove them because in that case, not so threatened. Also on a separate note, I just had a paper accepted on reference sites for restoration purposes, wetlands, not individual species, but it is about reference sites in highly impacted landscapes. So I might share that link here, or I can send it to Jill and she can pass it on maybe.
Yeah,
you know, it might be of interest. So I'll share that, Jill. Thank you.
Thank you.
We have Annika's comment in the chat on wondering if you've seen more involvement in any virtual program that you've done over the last year.
Yeah, so Budburst, the community science project I described, has been entirely virtual over the last year. We have created curriculum for people to use at home, whether they're you know, doing at home, either homeschooling or at home learning from other schools. And that's been incredibly popular over the last year because, you know, it allows people to do activities, particularly children, that get them outside, but also have learning goals associated with them.
Yeah, that's fantastic. Jess, you're next.
Sorry, hi. I'm terrible at asking questions during these because my brain takes my mouth takes time to catch up with my brain. But I was curious, a couple things. One was you showed your PhD students box plots of the commercial seed diversity compared to remnant, compared to restoration. And I was curious, was that all multiple restorations at different time points, or did you track one restoration and how that diversity changed over time?
It was multiple restorations, and I think they were all at least 10 years old. Not positive on the time frame, but it was multiple sites, but all fairly mature restorations.
Okay, and I guess so. Do you know if there was monitoring over time to see, you know, if there's like a peak time? Because obviously in the initial stages of restoration, which is what I'm looking at, it's a lot of weeds. So kind of curious of that trajectory of where we see the peak richness, and then if there's any trends and as that, you know, if there's a decline when that occurs.
You know, she didn't do that. But I do know there's been some chronosequence work, particularly at Fermilab in the Chicago area that has a really old restoration. And also Morton Arboretum, I believe, their prairie restoration is decades old at this point. So might be worth checking with them.
Yeah.
Becky Barak is wonderful. She's now on the faculty at Chicago Botanic Garden. So if you want to contact her and ask exactly what she did.
Yeah, awesome. Thank you. I have one more question if I could squeeze it in. I was curious, I'm interested a lot in regional admixture, and I know you mentioned I can't remember the exact name, but you know, going for what the future climate's going to look at, and you know, kind of using SDMs in that. Mm-hmm. And I was curious because we're now in 2020, and some of those early species distribution models were predicting to now, and I'm curious if this is also for me, if you've done any work that's kind of looked at, do some of those early models, do they actually match up with, you know, predicting to where we are now?
So we haven't. All our work was looking at 2050. Okay. So again, I'm sure there are people that have, but not us.
Yeah, no, that's one of those things where I was like, I should really start looking into that. So I just thought I'd check and see if you've done any of that. So, well, thank you so much. This is a really great talk and really applicable to my interests with the Hamilton Lab.
Wonderful.
And I think we have another question from Kate in the chat. She has a couple questions if you have time. There's a large demand for seed and restoration. Where does the Chicago Botanic Garden stand on cultivation practices? And she's wondering what your thoughts are on capturing those early flowering forb seeds for large scale restorations.
So we recognize that cultivation or bulking is going to be necessary to create the amount of seed we need for restoration. We also have done some work on how to do that bulking most carefully. So there was a paper, Basin, B-A-S-E-Y et al., 2015 maybe, called 10 Rules for Producing Native Plant Material. I think it's in Native Plants Journal. And that really outlines how we take plants through the bulking step in a way that preserves the most diversity, starting at seed collection and going all the way through growing it in production fields. And capturing early flowering forb seed for large scale restoration. Yeah, it's critically important. Most of those species have been difficult in production and require a lot more hand holding in the production process. And because of that, they're not profitable. And so many nurseries don't want to carry them. And so this might be an example of the type of plant that we need some public support to get into restoration, and whether that's growing at a native plant material center or at a botanic garden or another nonprofit to make sure that we can increase the diversity of what's available.
Thank you. That was my question. Thanks for conveying it, Jill. You said that was Basin 2015?
2015, I think. I don't remember. Basin et al., Native Plants Journal, 10 Rules. If you Google that, you should find it. Okay, or I'll send it to Jill and she can pass it around.
Thank you. And thank you so much for speaking with us today.
It's been very fun being here. Got to go to North Dakota without ever leaving my bedroom.
Which might be the preferred way to do it.
Give us a few more weeks. It's going to get a lot better really quick. Craig, did you have a question?
Yeah, so some work by Sfacioluda on the plat thistle years ago looked at invasive weevils and their effects on that endemic in western Nebraska.
Yep.
They really hammered those invasive weevils were really.
Yeah, it's the same suite of weevils that's hammering Cirsium pitcheri. So we've been in touch with Savacha about them. And she predicted this, 25 years ago. She saw them moving into native thistles, and sure enough, she was right.
That's unfortunate. Are there islands that haven't been invaded where the thistle occurs?
There's a few in Lake Michigan, Beaver Island. They're kind of on the Michigan side. We unfortunately find them on Washington Island, which is off the tip of Door County in Wisconsin. So they've made it that far, and that's like a 7-mile jump, I think, from the mainland to that island, and we've made it. So these little weevils, they get around, and it's unfortunate.
So is that Beaver Island population of thistle, is it large, and does it have high genetic diversity more than you'd expect?
So I haven't been out there to look at that population. I have a colleague named Benny Girdler at Kalamazoo College that monitors it, and you know, she says it's in decent shape, but you don't want all your eggs in one island. So.
Right, exactly. So, I had another quick question. That big sagebrush. So the local population did best, but there are other populations that seem to do well.
Mm-hmm.
Were there any commonalities among them in terms of their success?
Yes, that wasn't our work. That was Alan Sand's work, and so I don't recall off the top of my head if you know the populations that had about 50% survival versus the populations that had 10% survival, if those kind of medium ones were nearby or similar environment. I don't recall.
All right. Well, thank you. I had one last question / comment. I'm sitting in on a working group regarding loss of temporal genetic variation. And most of the work's been done with animals. And the leader of that group, where there have been all these meta-analyses looking at loss of diversity since the Industrial Revolution, and it seemed plant collections would provide some good opportunities there. And I think they said there has been virtually no work done on plants. So first off, if you're interested, I can put it in the chat. I've been going to this, it's every other Thursday. There's a temporal genetics working group. It's really, so far it's just was a seminar last week, but do you have any comments or interest in that?
Yeah, sure. Send me the information because it may be that we have data that's quite relevant to that.
Yeah, cool. Thank you. A very nice talk.
Thank you. Nice meeting you all.
Nice meeting you.